Hi everybody, let's talk now about the cytoskeleton of the cell. As a reminder, we're in Chapter 1, Section 1-2 of the Biology Kaplan Review book. Okay, so the cytoskeleton provides structure for the cell and also helps it to maintain its shape. In addition, the cytoskeleton provides a conduit for the transport of materials around the cell. I'd like to think of it as a kind of highway system. There are three components of the cytoskeleton: microfilaments, microtubules, and intermediate filaments. Let's talk about microfilaments. Microfilaments are made up of solid polymerized rods of actin. So actin is the protein that makes up microfilaments. The actin filaments are organized into bundles of networks and are resistant to both compression and fracture, providing protection for the cell. Actin filaments can also use ATP to generate force for movement by interacting with myosin, such as in muscle contraction. Microfilaments also play a role in cytokinesis, or the division of materials between daughter cells. During mitosis, the cleavage furrow is formed from microfilaments, which organize as a ring at the site of division between the two new daughter cells. As the actin filaments within this ring contract, the ring becomes smaller, eventually pinching off the connection between the two daughter cells. So imagine you had a balloon filled with water, and you took a rubber band or a piece of string halfway around that balloon and slowly pinched it off, so much so that you suddenly have two separate balloons. That's kind of a visual image for the process of the cleavage furrow and cytokinesis. Remember, actin filaments within this ring contract to form those two new daughter cells. Microtubules. Unlike microfilaments, microtubules are hollow polymers of tubulin protein. So remember the difference. Microfilaments have actin, and microtubules are made of tubulin proteins, and they're hollow. Microtubules radiate throughout the cell, providing primary pathways along which motor proteins like kinesin and dynein carry vessels. Cilia and flagella are motile structures composed of microtubules. Cilia are projections from a cell that are primarily involved in movement of materials along the surface of the cell. For example, cilia line your respiratory tract and are involved in the movement of mucus. Whereas flagella are structures involved in movement of the cell itself, such as the movement of sperm cells through the reproductive tract. Cilia and flagella share the same structure, which is composed of 9 pairs of microtubules forming an outer ring, with 2 microtubules in the center, as shown in the figure in your book, figure 1-5. You can also Google or search for this image on the internet. This is known as a 9+2 structure, and it's seen only in eukaryotic organelles of motility. Bacterial flagella, which we'll learn about later in this chapter, have a different structure with different chemical composition. The book would like you to think about this bridge concept. Motor proteins like kinesin and dynein are classic examples of non-enzymatic protein function, along with binding proteins, cell adhesion molecules, immunoglobulins, and ion channels. Motor proteins often travel along cytoskeletal structures to accomplish their functions. Non-enzymatic protein functions will be discussed in Chapter 3 of our biochemistry review. Moving right along, centrioles. Centrioles are under the header of microtubules. Here's why. Centrioles are found in the region of the cell called the centrosome. They are the organizing centers for microtubules, and they're structured as 9 triplets of microtubules with a hollow center. It really helped me to look online at an image, you guys, in order to fully understand the concept of centrioles function and its relationship to microtubules. So I recommend that you look up an image of this. Again, centrioles are found in a region of the cell called the centrosome, They are the organizing centers for microtubules and are structured as 9 triplets of microtubules with a hollow center. During mitosis, the centrioles migrate to opposite poles of the dividing cell and organize the mitotic spindle. The microtubules emanating from the centrioles attach to the chromosomes via complexes called kinetochores, and can exert force on the sister chromatids, pulling them apart. Finally, the third component of the cytoskeleton are intermediate filaments. Intermediate filaments are a diverse group of filamentous proteins, including keratin, desmin, vimentin, and lamins. Many intermediate filaments are involved in cell-to-cell adhesion or maintenance of the overall integrity of the cytoskeleton. Intermediate filaments are able to withstand a tremendous amount of tension, making the cell more rigid. In addition, intermediate filaments help anchor other organelles, including the nucleus. The identity of the intermediate filament proteins within a cell is specific to the cell and tissue type.